So bioinformatics for infectious diseases is really a program designed to address the pressing need to understand, characterize, monitor, and address by intervention the infectious diseases that are so prevalent and are characterizing this 21st century. So in this program, we'll talk about how to apply data analysis tools, and a lot of them are going to be somewhat similar to each other, but applied in very different types of outbreaks, epidemics, coronavirus, Ebola virus, rhinovirus, tuberculosis, malaria. So different types of viral, bacterial, and other pathogenic diseases. The program is really designed in a similar fashion. You will explore relevant topics, tools, methods, data repositories, project examples, and you'll be able to see how multi-omics analysis of genomic, transcriptomic, proteomic data is going to inform different types of decisions about outbreaks. Before we jump into this, let's briefly talk about what is the definition of infectious diseases. So infectious diseases, also called communicable diseases, are caused by a pathogen, and that pathogen can be a bacteria, virus, parasite, or fungi. And these kinds of diseases are different from non-infectious diseases, or what's commonly known as chronic diseases. And so we will actually talk about specific applications of bioinformatics to the study of infectious diseases. Now, in the last couple of years, all of us have been focused on COVID-19, and I think really as a result, we kind of see infectious diseases primarily caused by viruses. But in reality, they could be caused by different types of pathogens, including bacteria that can colonize and kind of subdue different types of functions that are important, but also can release toxins and cause organ failure. So in the recent years, there have been a number of pathogens like the Ebola virus in 2014, the outbreak that brought to the forefront genomic surveillance of epidemic outbreaks. There have been diseases that have been affecting millions and millions of people. And we see that these diseases actually are not just the ones that are new, like novel coronavirus, but there have been for centuries, like malaria. And malaria affects a huge amount of people and has been for many, many decades or even centuries. Novel diseases caused by the coronavirus or by re-emergence of specific diseases are going to continue and be very important and dangerous situations that we'll have to address using such tools. Now, importantly, these kinds of outbreaks are only one type. They are kind of silent, and then all of a sudden there's an outbreak. But there are many viruses that cause or lead to chronic diseases. And so even though we kind of tend to separate these two categories, there is a quite large overlap. For example, Epstein-Barr virus and other types of viruses can lead to cancer. So in addition to that, right, beyond viruses, there is a whole field studying different types of bacteria. And for example, drug resistance, microbial or resistant to antibiotics is a huge concern. And so in this program, we're going to talk about examples in all of these different fields. And importantly, we will also talk about the response to these diseases that our body already possesses and how different types of treatments like vaccines are leveraged to actually enhance this kind of a response. So when we look at the history of infectious diseases and we look at the methods to control such outbreaks, you will be surprised to find that actually vaccines that we talked so much about today are not the only ones that were responsible for the reduced load of infectious diseases on the population. So from population health perspective, we can see that the establishment of health departments, municipal use of chlorine in water, use of penicillin are all predecessors of the vaccines, which was introduced in the 1950s. And up until today, those methods were actually very, very effective. However, what happened just recently is all of a sudden, After having such success with methods that we already discussed, COVID-19 has surpassed the mortality of even such diseases like cancer. And so on one hand, we have this dynamic where we have effective measures that are quite simple. Just washing your hands, having chlorine in water, having availability of antibiotics. And then all of a sudden, we see this tremendous growth of mortality caused by a virus that has already been studied. And so that leads us to the significance and the impact of bioinformatics. So in the era of COVID-19, not only were we able to analyze that data quite rapidly, but very quickly we were able to identify a specific pathogen that then was tracked almost in real time to understand how it changes, how it evolves, how it starts infecting more and more people, and starting to ask important questions. And the ability to ask those questions using data that was collected is the essence of bioinformatics. What is bioinformatics? Bioinformatics allows us to leverage these data sets that have been collected, ask specific questions, and get to a practical interpretation so that we understand something and we're able to make conclusions. Now, importantly, this pandemic has opened a number of new questions that are now being asked. And we will see how those questions are being asked by researchers that have the expertise to design experiments or to leverage data that was collected from others. So here's an example where we can see that we can not only detect a specific strain of a virus, but we can actually track it over time, and important questions will start to emerge. What is the long-term consequences of COVID-19? How do we monitor and prevent outbreaks of these infections? How do we leverage our detailed genomic characterization of the virus or the pathogen and introduce a very specific targeted therapy that can prevent or kind of make it less dangerous? So with this brief introduction, I will again pass on the stage to Dr. Raghav to speak about the specifics and examples that are relevant for this upcoming program, and answer any questions that you might have about what are the outcomes of such training. So over to you, Dr. Raghav.